everyone, in this video we will discuss the question number 9 of GATE 2013. So the question is asking the nucleotide analog used in the DNA sequencing by chain termination method is, the four options are 1, 3, 1 prime 3 prime dideoxynucleoside triphosphate, 2 prime 3 prime dideoxynucleoside triphosphate, 2 prime 4 prime and 2 prime 5 prime. So now we will see that which nucleotide analog is used for the DNA sequencing. So if we will briefly discuss the DNA sequencing method by the chain termination reaction. So in this reaction we use a dideoxy DNTP analog, which is DDNTP analog, where you can see in this 2 prime position of the pentose sugar of a DNTP there is of course a hydrogen because it is a DNA, but at the 3 prime carbon also, instead of OH it is H, so that's why it is dideoxy. One deoxy here, another deoxy here, so that's why it is dideoxy. And this molecule, if it is incorporated in a growing DNA strand, then this molecule cannot be used for the chain elongation. Why? Because there is no 3 prime hydroxyl group, right? So that's why the nascent DNA chain will not be elongated, so DNA replication will be stopped, right? So now suppose we are given a template with sequence this one. So now we need to derive the sequence of the template, right? So for that we will first use the primer, which will be amplified by the DNA polymerase at the direction of 5 prime to 3 prime. So in this reaction mixture we will add all of the 4 DNTPs, and along with that we will make 4 different sections. In one section we will use DDATP, another section we will use DDCTP, DDGTP and DDTTP. So now let's see what is happening in the DDATP. So if we will add DDATP, so consider this sequence. So if we will add DDATP, then what will happen? The growing chain will start growing, and here, so if there is thymidine in the template strand, then this DDATP will be incorporated. So let's see. In the second position there is a thymidine, so one of the product will be GDDA, then the replication will be stopped. Another possibility of getting a product is another thymidine is here, so that will be GATTCGDDA here, right? There is another thymidine here, so for that case we will have the entire sequence C GATT, CGA, GCTG and DDA. So we will get 3 fragments. So if we will run it in a gel, then we will get 3 bands. One is the smallest band is for this fragment, right? For this fragment we will get the intermediate size band, and for this longest fragment we will get this upper one band, right? Similarly, if we will add DDCTP, then in the template strand if there is G, then this DDCTP will be incorporated here, and once it is incorporated, then the chain elongation will be stopped, right? So let's see. In the template strand there is a G here, so it will be GATT right? Another G is here, so the corresponding sequence will be, the complement sequence will be GATTC GAC DDC, right? So here the chain will be terminated. So these are the 2 possibility of product after addition of DDCTP. So right, so this band corresponds for the smaller product, and this band corresponds for the larger product, right? Similarly we will get DDGTP. If we will add DDGTP, then if there is a cytosine in this template sequence, then we will get that number of bands. Similarly the thing will be same for the DDTTP. So now if we will run the gel of DDATP lane, DDCTP lane, DDGTP lane and DDTTP lane, then the way to read the template sequence is, first we will start from the lowermost band, right? So the lowermost band is here in the DDGTP lane, right? So in the 5 prime to 3 prime direction, the first sequence in the complementary strand will be G, right? Next we will see, next if you will go to the, if you will go in the ascending order, then the next sequence will be this, right? Sorry, the next band will be this, which is in the TD ATP length, right? So the 5' to 3' direction of the complementary strand, the next DNA sequence will be A, right? After that, these two bands come. So these two bands are from the DD TTP length. So the next two DNTPs are T and T. Similarly, the next will be C. After that, G will come, then A will come, then again G will come, then C will come. After that, T will come, then G will come, and at the end, the highest or the larger fragment, it comes on the A, DD-ATP length. So the last sequence will be A, right? So the last nucleotide would be A. So that is, so after reading this gel, we derived the sequence of the complementary strand in the 5' to 3' direction, that is GATT CGA GCTGA, right? Now to write the actual template sequence in the 3' to 5' direction, we will just complement it. So the sequence will be C T A A C G C T C G A C T. If you will check, it is exactly complement of this sequence, right? So to derive a sequence of a template at the 3' to 5' direction, we will first derive the sequence of the complementary strand at the direction of 5' to 3', and then we will just complement it, right? So after complementing the sequence, we will get, that will be our actual template sequence. So in this entire chain termination method, the DNTP analog we use, that is that is 2', 3' dideoxy. Why? Right? Because here there is hydrogen in the 2' C2 position, and there is hydrogen in the C3 position, right? So the answer will be 2'. So the answer will be the option B, 2' to 3' dideoxy nucleoside triphosphate.